So a lot of you are on a 1-hour schedule, so I'll get going early, a little on time, close to on time, and I'll try to get done in an hour. If not, I understand people have to leave, and you won't be being rude if you get up and leave at 1 o'clock. If you have questions and have time and want to stick around, I'll be happy to answer questions. I can answer them during the presentation, but it'll slow us down. So, but if I'm very unclear about something and you want to ask me a question, please go ahead and do it. Let's just try to get through it. My name is Lenny Rome. I'm the Research Dean of the School of Medicine. I've also been a faculty member here in the Department of Biological Chemistry for about 30 years. That's where all these gray hairs came from. And I've been turned down on so many grants that makes me an expert in getting funded. So that's probably why I'm giving this talk. The talk today is going to be organized around getting funded, writing grants, mostly focusing on the NIH. You'll see why in a minute. Some of the nuts and bolts of the grant writing process. So that's what my focus is going to be on. But I'll start out with some more general statements about it. The hard reality that I start out with is to just tell you that about 10 or 15 years ago, one could expect to apply for their first NIH R01 being the investigator-initiated grant, directly after completing a postdoctoral fellowship. Today, the average age for someone to get their first R01 is now over 40 years old. So it just shows you that it's getting harder and harder, and people are further and further along in their career before they get their first NIH R01. In 2005, the funding levels for NIH grants were in the 15 to 18% range overall, depending on the institute. For this year, it's expected that 10% or less of grants will be funded. And for the past 5 years, the funding levels at NIH have been stagnant. And if you factor in inflation, that means that actually in real dollars, the amount of money has been going down now for 5 years at NIH. So that's the hard reality, but we're going to talk about getting started. And since the audience is heterogeneous, there are people here from basic science departments, clinical departments, people involved in basic research in clinical departments, human subject research, clinical research. I'm going to stick to comments that are generally applicable, but there's going to be a lot of differences based on the kind of research you do. So I want to start by saying it's very important that you establish with your chair and your mentors just what's expected from you with regard to the amount of research you need to do to further your career, et cetera. And there is different requirements for different faculty. So you need to understand what's expected for you. You need to set realistic goals. You should be very aware of the fact that you will not get your first NIH grant that you apply to, okay? It's much better to focus first on some smaller grants. We'll talk about other places to look for money to try to get some preliminary results. We'll talk about the importance of that. You want to be focused on what your goals are and make sure they're realistic. Otherwise, in this climate, you will be disappointed. Explore your opportunities. And what I mean by that is, if your number one interest in research is to study the structure of the large toenail, and you go out there and find out that there is not one agency in the world who is interested in funding research in the structure of the large toenail, you better get another number one interest, okay? It's going to be a hard sell for you as a junior faculty member or starting out to convince people that, that this passion is something that they should fund. So I don't exactly mean that you should tailor your research around where the most money is, okay? But if it just so happens that your research could be done in stem cells as opposed to normal cells, and just knowing that there's $300 million a year of California stem cell money out there, it might be a wise idea for you to concentrate first in a stem cell area, okay? So look out there, know where your opportunities are, be realistic about getting funded in places where there are opportunities. Get ready early. I can't emphasize enough how important it is that you have written your first grant proposal about a year ago, okay? So that you have time to get feedback. And the important feedback is that you can take advantage of your colleagues around here who can give you internal reviews, okay? Since the likelihood is that you and me, and I've been funded for 30 years, I'm going to get shot down on my next NIH grant. I mean, that's the likelihood. So I'd much rather get shot down by my colleagues first and understand where the problems are, and so that my first submission is really a second submission or a third, right? Because I've gone through a lot of the pitfalls, and that's incredibly important. I'll repeat it. I'm not going to spend a lot of time on this slide, and in fact, I give you more information on the slides than I have time to talk about. That's so if you go back, you can get more details. And I'm going to just touch on some big ideas in some of these slides. And the point of this slide and the next one is just to say to you, understand what it's going to cost to do your research. It's not always obvious when you're starting out. You think, oh, well, how much does a technician get paid? And, oh, I can hire a technician for $29,000 a year. Well, then you find out, well, you know, with benefits, that can be $45,000 a year. That might eat up a lot more of your grant than you want. So you have to understand the costs involved. A graduate student costs $27,000 plus tuition, for example. Supplies can be very large depending upon whether the kind of experiments you're doing. If you're doing animal experiments, okay, you can just double your expectations of what the supplies are going to cost and the animal costs are going to be, because it's very expensive. If you're doing health services research, then you're not spending as much money on supplies. So there's a lot of things you need to know about. Equipment, you're not going to get a lot of equipment funded in grants, okay? Maybe some small equipment, but they're going to expect that you've gotten, you know, you're from a big university, that you've gotten set up and you have most of the stuff. Large equipment you can get by going into shared instrumentation grants eventually, or here I would advise you to be taking advantage of our core facilities. Okay, again, we'll talk about that a little more. I give an introductory lecture to sources of funding to all new faculty, so probably a lot of you have heard that, and this isn't the same lecture, but I want to have an overlap, and on the first page of the handout, if you didn't get one, there's a few up here. On the first page of the handout is my web address, and there are a lot of ties in on the web on the School of Medicine research website for places to look for funding, okay? And the major sources of funding is of course the NIH, and in fact, if you look at the amount of money that's out there for funding biomedical research, the NIH is so much bigger than everyone else that's why I'm going to focus on the NIH grant today, okay? NIH's total budget's $32 billion. The next biggest funder of any research is NSF, and that's $7 billion. And then after that, the biggest foundation that gives out research grants is probably American Cancer Society, and that's only about $100 million a year, okay? So it drops off pretty rapidly. But I'll show you some of these other areas. What's really good, though, are for you to concentrate on small foundations that might be interested in your particular area of research. So, for example, if you work on cystic fibrosis, there's the Cystic Fibrosis Foundation. If you work on multiple sclerosis, there's the MS Foundation, etc. Most of those foundations actually have small grant programs for junior faculty to get started. And you're at a stage where you should be taking advantage of any kind of edge you have for getting started kinds of grants, okay? Even small amounts of money that let you get some preliminary results are well worth pursuing, because it's going to be very important that you have some results to take to NIH when you go for a bigger grant. I mentioned the California stem cell. California also has an initiative that puts money into tobacco research, prevention, etc. So there's aspects of that you can pursue. There's training grants, there's industry grants, money from industry, and of course, if you're in clinical departments, you have some access to private donors, and there are quite a few intramural programs. Again, if you haven't heard me talk about them already, go on to my website and look up the large variety of intramural programs we have for UCLA faculty to apply for grants. Okay, I want to just briefly go through the alphabet soup of different grant types that exist at the NIH, because it's so confusing for people that are just getting started, okay? So the thing to understand is that there's these categories of grants, and the one that you're going to be most interested in are called research project grants, or RPGs, And the big one is the R01 research grant, which is also called the investigator-initiated grant, etc. It's the sort of sine qua non grant that you want to have as part of your career to be a NIH investigator. But there are other ways to get into research project grants, such as the R03 small research grant and the R21 exploratory and development grants. And what you should be looking for is that often agencies put out calls for proposals, so-called RFAs, that are in specific areas, and often those calls are for R21s or a combination of R01s, R03s, and R21s. And if you're working in a specific area that's called for proposals, that's a wonderful opportunity for you to talk to the program officers at NIH that are in charge with that program and find out whether or not you fit a niche within that program that would give you an advantage for applying to a grant. Given that it's so hard to get grants, and I showed you the statistics, you're upping your chances by responding to a call for proposals, because usually a special study section will be put together to review those proposals, and they'll just be looking at proposals in that call, in that area. Okay, so it can be an edge. But there are all kinds of other R grants, so don't get confused when you see them. R15s, R13s, R41s. There's just lots of different R grant categories. There's fellowship awards that come under the F grant types. F30s are predoctoral fellowships. F31s are individual fellowships to predocs. There's a whole variety, but that's where this nomenclature comes from. Those of you that are in clinical departments who might be working on K awards already know about K awards, but you might not know that there's a half a dozen or a dozen different categories of K awards that are some good for young faculty, some good for mid-career faculty. There's a new one called the K99 or ROO, Pathway to Independence Award, which is for postdocs to get 5 years of funding that they can carry to their assistant professor position, even at another university. It's a brand new grant type. I'm curious to see how that's going to work out, but it's a new type because NIH recognizes that it's hard for assistant professors to get into the system, and they're hoping that this is a grant type that will get people into the system. There's a whole bunch of other alphabet soup awards, P awards. I want to point out the P01 because if you're lucky enough to have a collaborator that's a big shot researcher, okay, who's running a big program here, they might have what's called a P01 award or a program project grant. That's usually a grant that's made up of 4 or 5 R01 type grants put together on a common theme. And for a young faculty member, it's easier to get integrated into a program project grant and get funded the first time as a project in a program project grant, as opposed to being a completely independent grant that you've put in sort of cold turkey on your own. So if you're fortunate enough to be able to partner with a group that's already well funded, and this can be a competitive renewal of a PL1 grant because projects come and go. Those grants are very heavily scrutinized by NIH to make sure they stay current. So it's to the PI's advantage to be bringing new blood, new projects into their program project grants. So it's a type of grant you should definitely be paying attention to. And it's a great way to get into the system because you can get almost as much money as an R01. You can get it as a part of a big project. If it takes off and it's successful, you can apply as a competitive renewal. You can move it into an R01 series. So there's lots of things you can do with a P01. So I wanted to point that out. I also list some specialized centers, the shared instrumentation grants, all kinds of different awards here that are coming and going. All these are summarized and described on the NIH website, and I've given you the link on this slide as well. Yes?
Is the P20 sort of the equivalent of R21 versus R01?
Yeah, they can use it that way, where a team can get an exploratory grant. I believe that's what it's used for. Another one that's interesting, that's getting more popular, is this U01. It's like a program project grant that the NIH is allowed to micromanage. So it's like they can be bugging you every 6 months for progress reports, changing the personnel on your grant, really focused on results, okay? Like the DOE does and the DARPA, where there's real hands-on from program officers. I think NIH is getting under the gun to get more results, and so they're getting more serious about it. So again, links to how you get information. This is just one slide, but go to my website, and there's a lot of links to all the local agencies. Any calls we get, we link it to their local websites. The best place is to stay current by going to the websites of the people offering the money, and I've given you the big links on this page. So getting on with deciding how you want to sort of further your career in research, it's really a good idea, like your stock market portfolio, it's a good idea to diversify, okay? And if you can have grants from small agencies, large agencies, private donors, federal donors, if you can mix this up, you're protecting yourself, because certain sources come and go, and you don't want to leave yourself vulnerable where your whole lab is depending upon one grant that maybe doesn't get competitively renewed. These are tough times, and it's I see it happen all the time. I see faculty who are National Academy of Sciences members, okay, get unfunded because of the NIH system, okay? So you are not alone in this tough times of getting funded. But the more diversified you are, the more you're getting money from multiple sources, the more you're going to be able to buffer those hard times. So it's something to think about. People talk about the Howard Hughes Medical Institute, but for you, I just want to make it more of an information item. The Hughes has a huge endowment, $14 billion, but they don't give away most of it to competitive grants, okay? They give away most of their yearly endowment, $730 million a year, to support just 375 investigators who are called Howard Hughes Medical Investigators. We have several of them here at UCLA. These are the superstars, okay? You should all be aspiring to be a Howard Hughes medical investigator, but you can't apply to be one at this moment, okay? This is, and the other money they give away is also very targeted, so they don't really have a program like an R01 program that like the NIH has, okay? So you should be aware of that. However, there are hundreds and hundreds of private foundations out there, and I've just listed the top 20, which have $500 million a year in grant funding, just the top 20, and over 600 grants a year. So this is real, and if you add up the foundations just available to you, it's billions of dollars and thousands of grants. So it's a source that you should be thinking about. Finding the right ones that fit your research is very important. So I talked about the NIH spending. NIH is the real big dog here, and they have a budget proposed for 2011 of $32 billion. Where is that going? Well, you'll be happy to hear that 53% of it is going into these R grants, the research project grants. And the pressure has always been on the NIH to fund R01s as their major source of grants, okay? But R01s, R03s, R21s make up the bulk of NIH funding, okay? These other pie pieces also affect you. For example, research centers like our cancer center are funded out of the NIH on center grants, which can indirectly affect you because of the core facilities and the things that centers supply for research. It's also curious that about 10% of the NIH budget is intramural programs, okay? So it's pretty nice to be at the NIH having 10% of the total budget of the federal government being spent intramurally. The NIH money, though, is not divided equally. It's spread out in budgets of all of the various institutes of the National Institutes of Health. And the biggest institute that gets the biggest share of the pie, which isn't even a quarter, is the National Cancer Institute, okay? And you can see all the various institutes marked here. Some of them get such a small slice I didn't even put their names down, okay? But there's still a lot of money here. And the funny thing is that each of these institutes often has a different cutoff range for funding, okay? So it does sometimes matter what institute your grant's assigned to, and I'll talk about that in a little while. Yes?
Is there a universal NIH salary cap, or does it differ based on the institute?
I believe it's a universal number for the salary cap. It's not institute-wide. So the grant, I'm going to focus on the NIH process because, as I said, if that's where most of the money is, almost all of you are going to be thinking about or trying to decide how you're going to get funded by NIH. So the rest of my talk will be mostly focused on the NIH process, on how it works, and on my suggestions for you in terms of writing grants to the NIH. Okay, so how it works is now, and everything's changed in the last couple years. The last competitive renewal I put in was on the old system because I was grandfathered in because it was a competitive renewal. So I haven't even had my grant go through this system, although I'm writing it in this way now. But so this is all new, and so we're learning new experiences about this system. It's all electronic now. In the old days, everybody was scrambling around to make all their copies, and I had colleagues that were so late that they actually had to jump on an airplane with their grants in hand and take a taxi from the airport in Washington to the NIH so that they could get their grants delivered on time to the agency. It was not unusual for that to happen. Okay, now it's electronic submission, and what you have to worry about is the system crashing on the day that you're delivering the grant. So they tell you here to try to be a day early so they don't get involved in this enormous amount of web traffic that's going to go on at the last minute. Okay, so it goes to a place called the Center for Scientific Review, and they read your abstract and a cover letter if there is one there, and they decide what of the institutes they're going to assign your grant to and what study section they're going to assign your grant to be reviewed by. It's a split process, okay? A single study section will be reviewing a group of grants in a topic area, and they may be assigned to all different institutes, okay? So the institute directors that are involved in the research that are interested in those grants come and sit in on the study section when they meet. So they have an interest, but they're not reviewing the grant, okay? So when you see your assignment, you want to pay attention to the assignment, because in some cases, your research you think should be in the Heart and Lung Institute and it got assigned to cancer, and you might have a good argument that it's been assigned to the wrong institute, or that they should dual assign your grant to 2 institutes, which actually increases your chances of funding. Because if you're right on the border, one institute may fund at a better rate for your grant than the other. And so you want more than one institute interested in your grant so that the one that has the more money can pick you up. Okay? So there is some politics here. A lot of the politics is area specific, so I'm going to not spend a lot of time talking about these politics, except a little bit I'm telling you about. I want you to be aware of them, and your mentors are good places to go to in terms of how much should you be talking to program officers, how much should you be talking to execs of study sections. There's a fine line between schmoozing your executive secretary of the study section and getting information, okay? And you should be aware that they get called by everybody, and they'd rather get called when there's real issues, real questions coming from you. Then you can use the opportunity to make sure that they know you're a really good guy, a really nice person, a really good researcher, and they should look out for you, okay? But you can't call them for that purpose, okay? Your grant gets reviewed. I'll talk about the scoring process. I'll talk about the criteria, et cetera. It then goes to a council. An advisory council is like a higher body that can pull up some borderline grants. I don't believe they ever take a grant that's in the funding range and take it out of the funding range, okay? But they can bring grants that are not in the funding range into the funding range, and for programmatic reasons, okay? So it can affect marginal proposals. Then there's a financial review, and then there's an award. All you care about is that you get this letter that tells you we can't make any definitive conclusions to you at this time, but you're in the funding range. It's likely you'll get funded. You get this kind of letter. It has to go through the council. It has to go through financial review, et cetera. So, don't start spending the money yet. But if you get that kind of a letter, you can call up your contract and grant office, and you can actually start spending the money now, even before it's here, if you can assure the university that you are going to get funded. So there is a way to even get started early without the money being here. So you should know about that as well. So the grant, as I said, the proposal is directed to a panel of experts. This is the study section. They have diverse backgrounds and interests. And 3 reviewers in your study section will be asked to be primary reviewers on your grant, and usually three will be asked to be secondary reviewers. That means of the study section, and there can be 15 or 20 people on that study section, there's only 6 people that are going to read your grant, that are required to read your grant and write stuff about it, rate you on these criteria we're going to talk about. Everybody else is supposed to look at all the grants, but if you're on study section, these are people just like you that are overworked, very busy. They put off this stack of grants they have to read until the week before, or on the airplane when they're flying over. No, they can't do it that way because they actually have to submit their comments early. But they put it off to the week before, and they're in a hurry. So we're going to talk about strategies that are considerate of the reviewers, because that can help you as well. So your proposal must inform and persuade these 6 people, okay? And they may not be world's experts at your research, okay? Now they have to be confident enough that they can review your grant, because the study section exec secretary knows what they do and assigns based on their expertise. And if you work in an area that you don't think is covered by the study section, again, I'll touch on this later, you can try to influence the study section to bring an expert on board to review you. So that's possible as well. You have to show it's important for science, especially that it's important for human health. The NIH is the National Institutes of Health, and they are not interested in funding research, great research, that is not related to health, okay? They'll say, this should go to the NSF, which wants to fund great research that, might just affect a fruit fly, okay? So, if your work, if you can't show that your model system fruit fly work is going to be relevant to human health, don't bother. It'll just be triaged, okay? You won't get reviewed, all right? So it's very important that you focus on health. It has to be feasible. You have to show that you and your team can do it, and you have to justify the budget. This is how the scoring goes for each of the sections I'll talk about in a minute. The scoring is from 1 point to 9 points for each of the people scoring, 1 being exceptional to 9 being poor. But guess what? Half of the grants aren't even scored, okay? So you get, all the reviews come in early for the study section, all the study section people come together. They've each scored all of the parts of their grants. Okay, and all that's tallied before the meeting, and the executive secretary looks at the tally and says 50% of these grants are not going to make it based on your scores. Let's triage all of them and not even talk about them, okay? Unless somebody on the committee objects. Wait a minute, I read that grant, it's the greatest thing since night baseball, okay, and you guys need to push that up into the discussion phase, okay? It's gone, and you get the dreaded ND, not discussed, okay? You get to see your comments, okay, but that's a problem, all right? We'll talk about that when we talk about resubmission, okay? Now, if you're part of the 50% that gets scored, you still might get a terrible score, okay? You still might get a great score that's not even funded, okay? All right, so it's a tough system, all right? There are some other scores, but again, they're pretty rare. For example, if you propose some human research that they think will kill people, okay? They will score you as NR, not recommended. That means you can't even come back, okay? Don't even come back with that grant, all right? You certainly don't want to have an NR, all right? So let's talk about the review criteria, because this is what you're getting scored on, okay? Significance, the investigator, innovation, approach, environment, and overall impact. So we'll take them one at a time. Significance. Does the project address an important problem or a critical barrier to progress in the field? If the aims of the project are achieved, how will scientific knowledge, technical capability, and/or clinical practice be improved? I want to pause on this one for a minute, because many reviewers use this criteria. And what they say to themselves is, okay, let's assume that they accomplished every goal in their specific aims. So what? Okay? You better be able to explain to the reviewer why it's so important to furthering health, to furthering research, to furthering the paradigm, to furthering your hypothesis, that success of your aims will achieve. Because if your grant is very doable and they think you can achieve the aims, but when they look at the results and think that it's going to be a so what, you're never going to make the cut, okay? How will successful completion of the aims change the concepts, methods, technologies, treatments, services, or preventive interventions that drive your field? It's a very general statement because I'm talking about all kinds of grants here, right? But if you're talking about a new method, you better be showing that if you succeed with your grant, that's going to alter the way people do this methodology in the field. Investigator. Is the PI and the collaborators well suited to the project, okay? Again, it's a little tricky here. If you have wonderful training as a virologist and you're in a virology department and you've published great virology papers, okay? And then you submit an R01 to the National Cancer Institute to cure lung cancer, okay? Okay, and it's all about, signal transduction in tyrosine kinase. The reviewers are going to sit there and say, I don't see how this virologist has what it takes to do this complex signal transduction research in cancer, okay? So if you're changing fields or if your background and expertise does not predict that you can be successful in the grant that you're applying to, you better work hard convincing your reviewers that you can do it, okay? Letters from collaborators here at UCLA who are experts in the field talking about how you're uniquely suited to do this work because X, Y, and Z. Okay, other factors like that, okay, collaborators. Right? If early stage investigations or new investigators or early stages of independent careers, do they have appropriate experience and training? Again, these are not big problems for this audience, okay? Faculty at UCLA got here, were one of the top research universities in the United States. All of you got very competitive jobs. This is more for, this is actually an advantage for people at universities like UCLA, the investigator part and the environment part. I'll talk about in a minute. Nevertheless, you should be putting up the right data there for your reviewers to see that you're special. If you're an established investigator, do you have an ongoing record of accomplishment? You can't just ride on the fact that you're a full professor. You have to show them that you're continuing to work in your field. If it's a collaboration, they want to see complementary, integrated experience. They want to see leadership. Innovation.
I have a quick
Yes,
question about
go ahead.
investigator. So for the new investigator series in an R01, is there an expected number of first author publications
No.
you have in your field?
No, there's not an expected number of papers. There's even a stated, there's a statement by NIH that you don't need preliminary results if you're a first-time investigator. Don't believe that. We'll talk about that later. Innovation. Does the application challenge and seek to shift current research or clinical practice paradigms by utilizing novel theoretical concepts, approaches, or methodologies, instrumentation, or interventions? Are the concepts, approaches, or methodologies, instrumentation, or interventions novel to the field, to one field of research, or are they novel in a broad sense? Again, if you're very focused on an area of research that is very specific and maybe only affects some very small number of potential patients, and you're going to do some paradigm-shifting research, maybe they're worried that that really isn't enough innovation to carry the day, maybe. If it's a refinement, improvement, or new application of a concept, how will it improve the area? Okay, again, they really want you to tell them what's innovative about your research, because they're going to score you on innovation. By the way, innovation came about because one of the biggest things that NIH over the years has been critical for in the old system was that they tended to fund grants that were safe, doable, where people had already done the research, as opposed to innovative grants. And so they've moved innovation up, and the new system is supposed to reward innovation. And it's early, so it's hard to tell if that's really going to be the case or not. I hope that it will be the case, because I do think that when you get down to a small number of NIH grants being funded, you don't want just the safe grants to be funded. You want the research to be driven by innovative proposals. The approach is the most important part. It's really the guts of the research proposal, and so I'll spend most of the last 20 minutes talking to you about writing the proposal, which is writing the approach section. And again, I won't go over all the ways they evaluate the approach. It'll be obvious, so we'll save a little time talking about approach. Environment's another easy one, because they're worried, do you have the right environment to work in? And again, anybody at UCLA or our affiliates who's applying for a grant that can't nail the environment part, really it's just a grantsmanship problem, okay? If you're at Southern Eastern Illinois Catholic College, okay, and they have one EM that they bought on eBay, all right, and you're proposing to do a cryo-EM reconstruction, they're going to really be worried that you don't have the tools to do that work, okay? That shouldn't be a problem here. We have phenomenal core facilities. If there's any instrumentation that you need and it's not here, I'd be surprised, okay? You have to be able to convince the reviewers that you're tapped into this wonderful resource called UCLA, and that you're aware of and have letters from the core facility directors who are going to help you use instruments that you haven't worked on before, et cetera, okay? So again, the environment section, and again, go to your colleagues, go to your mentors who have gotten grants funded, and borrow language, because it's the same place, okay? It's not plagiarism if you find out stuff that they wrote about UCLA and the environment, okay? Of course, you can change the words to fit your grant, okay? But get help on this section, because we have terrific environment here, and you should be taking advantage of that, all right? Overall impact is very interesting. It is not an average score of how you score in the other criteria, okay? They look at all the scores and then they say, will the project exert a sustained, powerful influence on the research field? And that's the score that matters, okay? And it is not always even, close to an average of what you score in the other fields, okay? So, for example, if they really think you're innovative, even though you got some other problems elsewhere, they might boost you up and give you a better score than the average of your scores. If they really think that there's some serious problems, even though you've scored high in other areas, they might give you a lower score than your average, okay? So this is important. So common criticisms. Let's just plow through a few of these so we have time to talk about the writing. Significance is usually the big problem. It's this so what thing. If it's successful, is it going to change anything? Okay. So if the guy gets, if you get everything you said you were going to get in terms of results, does that matter or not? Okay. That's a big problem. Innovation, has it been done? Has somebody more qualified already doing it? You don't know this, but if you're in a very competitive area, that study section may have funded 2 Howard Hughes investigators and 1 Nobel laureate the time before to do the exact same experiments in a better way than what you're proposing, okay? It's not, you are in an area that is highly competitive if that's your problem, okay? You might rethink your approach if you're competing with much bigger guns with much more resources, okay? It can be a problem. Is it feasible? If you're doing population experiments, if you're a health services researcher, it's very important that you convince them that you can sign up the subjects that you say you're going to get, that you've statistically powered the experiments correctly. They're going to have experts at this, and they're going to be looking for reasons to not fund you, and they're going to find flaws in these areas. Is it focused? Do you have contingency plans? We're going to talk about that in a moment as well. Again, I've summarized a lot of the stuff I've been talking about on this page for basic guidelines, and we'll skip over this. You can read it in more detail. And instead, we'll turn to R01, and I'll try to give you a little bit of advice about writing your first R01. So first of all, like I said, it's a new system. It's described on standard form 424 or SF 424 R&R, which is found on this website at the NIH. There's lots of details about how to do this. I've summarized the whole thing in this one slide to just show you what's the important part here. I mean, you have to be aware of the number of pages, the font, all that stuff, how to electronically submit, what goes on administratively, what goes on in budget, what goes on in personnel, what goes on in resources and facilities, other things if you're doing human research like diverse populations, human and animal use and references, but the meat of your grant is 12 pages called the research plan, okay? That's what we're going to talk about. The research plan is divided up into 5 sections now: specific aims, significance, innovation, preliminary studies, and approach. As I said, you don't have to have preliminary studies if you're a new investigator, but in my opinion, you won't get funded unless you have new preliminary studies, okay? So we're going to talk about specific aims first. Specific aims is 1 page, okay? You want to market your application to the reviewers in this page, okay? You want to get them excited about reading the 12 pages that will follow so that they think, oh, this is cool. This might be innovative, okay? I want to find out how this person is going to do and be innovative and be significant the way they're describing here in this 1-page aims. It's a marketing, 1-page marketing brochure for your grant, okay? Briefly state the impact. Briefly state why you chose the project. Convey its significance and innovation, and do it in a way a broad audience can understand it. Because, like I said, many of those 6 people that are going to read your grant aren't experts at your research. They might be broad experts who are able to review your research, but you need to talk to them in terms that they understand. Choose specific aims that peer reviewers can easily access. Each one should be achievable endpoint rather than a best effort. What I'm saying by this is, and I'll talk about it more, your aims have to be well focused and they have to have goals that are achievable in them. And there can't be too many of them. I'll go into that as well. You should probably limit your application to 1 specific aim a year. That means a usual grant, 3 to 4 year grants, like 3 to 4 aims, okay? If you have a very ambitious project and you think it's very much driven by them seeing the future, okay, seeing where you're going to go once you do aims 1, 2, and 3, don't put that projection into aims 4, 5, and 6. Write a little future direction section and let them know where you're going if you succeed in this tight, focused grant that you're going to be applying for. Have no more than 1 or 2 sets of experiments per aim. We'll talk about that too. If you need lots and lots of experiments in your aims, you need to reorganize. Think of a specific aim as a framework of your application, an expanded table of contents for your approach section. And in fact, you want the exact same aim in your specific aim section to be the headings of the sections in your approach, in your 12-page approach that describes your experiments, okay? They have to be focused. They should have milestones. They should be testing hypotheses. So if you think about it as the specific aim sandwich here, you get 1 page, right? So I suggest maybe 1 to 2 paragraphs with a rationale, significance, and hypothesis. Then the meat, of the sandwich is these 3, maybe 4 aims, okay? And then a paragraph or 2 on why it's significant and innovative, okay? And then take those same aims, 1, 2, 3, and 4, and make them the headings in your approach section. So when you write significance, frame the problem and its general significance. Briefly review the literature. Interpret gaps in the knowledge. Indicate the terms in generally how you will fill the gaps. Show how the work is unique and new. Assess the potential impact on science and health. Back to health again. You must put that in your grant. okay? And finally, if it is a response to an RFA, then refer to the NIH priorities that are articulated in that RFA. Say, I'm responding to this particular RFA that asks me, okay, to understand the, how the appendix works in great detail, and that's my research, and that's why I'm applying here, okay? It's in the RFA, and tie to it. RFAs are sometimes quite detailed about what they want you to respond to, and the more you can tie your grant to what the NIH is asking for, the better you're going to come out in the review. Innovation. Briefly describe how the work is new, unique, and how it'll add significance to what's known. If your application is highly innovative, you'll need to build a strong case, because highly innovative proposals are asking to change the existing paradigms, and you're going to be reviewed by 6 people who are using the existing paradigms, okay? So you better be able to explain to them how this is going to work and how you're going to do this. And we'll talk about this more in pitfalls as well. Show how it's innovative. Present a broad view of the field and its significance. Now, the approach section, as I said, yes?
How many pages do you normally recommend for the significance and innovation section?
I think you get, I think the recommendation is that you do it in like a half a page or a page.
For each?
Yeah, a total. Yeah. So as you don't want to spend a lot of your 12 pages in the approach doing politics about, how great this is. I mean, you want to make, I've seen it done in a page of bulleted, innovative statements, okay? If this works, this is going to do this is going to do this. I'm going to change how people think about this, and it's just a focused set of statements, okay? You really want to spend most of your time, expanding on your aims, which is really the meat of the grant, okay? And you only get 12 pages, and that includes the significance and innovation part, okay? So sections have to correspond to the aims. You can briefly reiterate the significance within the sections. You should integrate your preliminary data into the rationale, the beginning of the aim, okay? That's where and you can cut it up. You can have a preliminary results section for each aim. You can have a preliminary results section that precedes the aims. You have a lot of flexibility. Look at grants that are coming from colleagues that are in similar areas and see what fits for your research. Present the logical structure, use multiple approaches, discuss pitfalls and alternatives, okay? You're going to say, I'm going to do this aim using the following methodology. If that doesn't work, though, I've got a backup plan, and here's my backup plan. You should always be asking yourself, what could the reviewer criticize about my grant? And then answer the criticism in your grant, okay? For example, if the first aim you have is a series of experiments that if it doesn't work, the rest of your grant is meaningless, that grant is dead in the water, okay? If there's a few experiments or one experiment that's going to determine whether the rest of the grant's feasible, you better have already done that experiment, okay? And it's a common problem that people have a creative grant and aim one, okay? It may sound great, it may be very logically laid out with a very good plan, but if the answer is no, and then there's no aim 2 and 3 after that, what are you going to do? I mean, that's like a fatal flaw, all right? You want that picked up by one of your colleagues before that goes to NIH, okay? Discuss how you're going to analyze the data and do interpretation. Some grant types, some areas of research are very much dependent upon how you're going to statistically approach the data, how you're going to have the right number of populations. I'm a basic scientist, and I always joke with the people in my lab that if I need a student t-test to tell me whether the experiment worked, it didn't work, okay? We're looking for a band on a gel, or a sequence or something, but that's not true of a lot of research that requires a very careful statistical analysis, and you better show the reviewers that you have what it takes to do that type of analysis, okay? Routine methodology can be lumped at the end. Now that grants are so much shorter, you really don't have time to do a lot of work in your approach section telling them what methods you're going to use or how you're going to do experiments. So the ideal thing would be to say, we're going to purify these proteins by the methods that we already published that we can purify these proteins by, and just put a reference in, okay? The best way to describe methods is to put references to your own work, okay? So that's really great. If a lot of the methods you're using are things that you've done before and you have a few publications about them, then don't spend any time proving to the reviewer that you know how to do it. Give them a reference that proves that you know how to do it, okay? Flow charts may be helpful. Pictures are worth 1,000 words, and since you have very little space here, it's a good idea to put some pictures in flow charts, even though you say, that limits the amount of words I can put in there to describe how great my work is. Again, it'll be kind to the reviewer. It'll help the reviewer see it. If you have a really good idea and a simple, focused plan, you can describe it with some nice, focused pictures and charts and data. Provide your own key data supporting the rationale. I said it's very important. Demonstrate relevant technical expertise that you can do it. Introduce, if you're doing this collaboratively, talk about your collaborators, what they bring to the table. Make sure you have reference letters from them in your appendix that says personally to you, oh, I'm so happy to be doing this research with you. I'm glad to share all the expertise that we've gotten in our lab in the past 25 years, all our specialized reagents. We're just going to give them to you so that you can be successful in this grant. And you know what? Your collaborators will write those kind of letters for you. They're glad to, and in fact, you need to draft them for them exactly the way that you want it to read, and then get them to give you that letter. Yes?
I have a question. One of my collaborators that I plan to invite, and we have several vacations together, and it will be very logical. Problem is that he's working in National Institute of Health.
Yes.
And they have some weird regulations about letterheads.
Yes.
And I read in Arrow 1 rules that they require them on letterheads. I mean, those letters of
Yes.
support.
So you should call your program officer on that one and get a clarification, because I know there are complications. They may require that you describe what your NIH collaborator is going to do for you, and they may accept that. That's one strange exception.
I need to describe it in a separate letter.
Well, you have to talk to your program officer to see if they will allow a letter to be added. You may just have to describe in your grant what they're going to do for you. That's one place where you're between a rock and a hard place. Yep. Okay. Point out special resources that distinguish your proposal. So for example, being at UCLA, if your proposal requires some really fancy technology that we are pioneering here at UCLA, you might want to point out why this puts you in a unique position to do this research. Okay? That's important. So again, with the sandwich approach, each specific aim might look like this, where you have a brief background, significance, hypothesis, rationale, strategy, and innovation that introduces the aim. And then you have the experiments, 1, 2, 3, no more than 4 experiments that is described in how you will accomplish this aim. And then finish it with, this is my anticipated results. Of course, there are pitfalls that could occur, and I've anticipated them just like you, Mr. Intelligent or Miss Intelligent Reviewer, have done, and I've thought about them, and I've decided that if this occurs, I'm going to do this, and if this occurs, I'm going to do this, okay? Put these alternatives down, okay? Think about the biggest flaws you have in your grant and state them before the reviewer does, okay? And state what you're going to do to make sure that that's not a problem, okay? It's very important. Then state at the end why that's significant, that result that you expect that you're going to get. Okay, helpful hints. We're wrapping it up, and I told everybody I'd be done in an hour. Make sure you use headings and subheadings that make it easy to read through your proposal. Make sure that your listed aims in the specific aim page are the exact same aims as you put into your approach section that you describe the experiments. Don't confuse the reviewer, okay? Don't get fancy. Don't make it more complex in the approach section than it was in the aim section. They're going to get lost. They're going to get confused. They're simple people, overly worked, just like you and me. They want to read a nice, clean grant, be convinced, become your advocate, okay? Let me take an aside here for a minute, okay? It's very unfair to the study section members what's going on now, okay? NIH peer review system works beautifully when about 1/3 of the grants are getting funded, okay? Because peer review is a very good way to take a bunch of grants and separate them into 1/3 great, 1/3 mediocre, and 1/3 bad. And peer review works terrifically when that's what the reviewers are asked to do. If they're then asked to take those 1/3 great, okay, and pick out the 1/3 of them that's going to get funded, they feel terrible. They've reviewed 6 or 8 grants themselves, and they realize that maybe one of them will make the cut, okay? Okay, you really have to give them something that they can champion, okay? That they're going to champion maybe 1 or 2 of the grants that they reviewed, because those are going to make the cut, okay? It's very tough. Okay, break up the text with figures, I said. Be really reader-friendly. You want to communicate that you're excited about doing this research, but you have to do it in a way that's realistic, okay? You want to close with a future direction section and a timeline. I'm going to get my aims done under this timeline, okay? So it's very good to have some kind of a timeline, either verbal or a chart if you have room, okay? And it's a very common problem that young, enthusiastic researchers really see beyond their first set of experiments, their first few specific aims. And they say, you know, if I can just get this done, just think about the next experiment I can do. And so they're so tempted to put that experiment in there so that people understand that excitement, okay? But then they're going to get criticized for overly ambitious, okay? And that's the number one thing that shoots down young investigators, okay? So you can never get criticized for being overly ambitious about your future direction section, okay? Okay, that's where you can say, if these work, you know, I'm going to have the cure for lung cancer, okay? That's where you can say that, okay? Because they can't criticize that him wanting to cure lung cancer is a bit of is overly ambitious, because that's not one of your aims, okay? That's where you're going in the future, all right? So, more advice, and I got it on your handout. Be careful how much you propose and how innovative you are, okay? Sharpen your application's focus. Again, novice PIs often overshoot their mark by proposing too much. Beware of being too innovative, okay? Tell you an anecdote here. The great Seymour Benzer, okay, you know, one of the most brilliant scientists of our age, okay, was at Caltech, recently died. He was here giving a talk about his work and how the first time he submitted his grant to NIH to use Drosophila as a model system for human neurogenesis, they laughed him out of the study section saying, how can you use fruit fly to study human behavior, okay? And, you know, that's how, you know, there's now fields doing this, right? But the first time, Seymour was too innovative, okay? Now, fortunately, he had lots of grants. He had diversified his portfolio. He just went back and did some model experiments and showed them that, yes, indeed, you can use fruit fly genes to model human behavior, and then the whole field took off, okay? But certainly on that grant, where he was essentially triaged, okay, it was because he was ahead of his time with innovation. So be careful about that. Common reasons for failure. I went through a lot of these. Your goals are nice, but they're not significant. You lack new ideas or original ideas. You haven't been able to demonstrate any accomplishment, even relative to your stage. That's why I said that for them to say you don't need preliminary results, you know, I think that's bull, okay? I think you need preliminary results. Weak scientific rationale, superficial or unfocused approach, lack of understanding of the literature, inadequate methodology, lack of experience in essential methods, illogical, disorganized, or presumptive approach, uncritical, scope is too excessive for the budget, resources, or experience that you have, lack of key experimental detail. Okay, must be very strategically supplied. Your senior colleagues basically put enough preliminary results in to convince the reviewers that they've just about got the grant done. Okay, it's a hard, unfortunately. The old system really was a system of rewarding people for already having done the work. That's where these criticisms of innovation came in, okay? So nowadays I'm hoping that with the new criteria, young faculty have more of a chance because they're more creative and more innovative than us older guys, okay? So that's important. If you get shot down, okay, hopefully you got scored, okay? Because if you got scored, even a bad score, you can bring that bad score up into the funding range, okay? If you got triaged, it's very difficult to bring a no discussion up into a funding range with one resubmission, so that's tough. Subsequent proposals may be screened for similarity. What they mean by that is you can't just get shot down twice and then repackage it again and say it's a new grant. I mean, you could get away with it if it goes to a different study section and it gets assigned to a different institute. That's okay. But if it goes back to the same study section, the same exec sec is looking at it, it better be different enough to be qualified as a new grant. Okay? Introductory page is used for direct rebuttal. So when you resubmit, you get one extra page in front of the approach section. So you have one page to rebut the reservations that were expressed by your reviewers, okay? And the best rebuttal that you could possibly have is data showing that it does work, okay? You know, if someone says, AIM-1 will never work, okay? They'll never be able to screen a mutant, you know, in that enzyme using that approach. If your rebuttal is, you know, we appreciate reviewer one and we agree, you know, we thought it was tough too, but maybe we got really lucky, but look, it worked, we got the mutant, okay? I mean, that's a great rebuttal to that kind of a thing. So you really don't want to quabble with the reviewers. They're all absolutely correct no matter how bad the review is. Even if they didn't even read your grant and, you know, scored you on some criteria from Mars, they were great. Thank them for the constructive criticisms and tell the new reviewer or the same guy what it is now that makes this grant fundable where it wasn't before, okay? Very important. The best rebuttal is new data. Okay, I'm ending here, but I know I've been depressing. I'm sorry, you know, it's a tough field. But these are tough times. Competition for research has never been greater. The amount of research funding is flat at the NIH, which means real dollars, it's going down. But I showed you there are still billions of dollars of research funding available that is being spent every year from federal government foundations, state of California, our university, the medical school, your department, okay? This money will be spent and those who are competitive will be funded. You guys are competitive, okay? You are coming from one of the top universities in the world, okay? UCLA was number 4 in total research dollars for the last like 3 years, okay? And we're always in the top 10 of NIH dollars, okay? So you just have to hang in there and you will get funded, okay? You are here at a really great place. I feel really bad for faculty who are not at really good places, okay? Because they have an even tougher time of doing this, okay? So I want to tell you that I'm sorry I went fast. This is all up on the web. We webcasted it, which means you can go back and slow it down and play me in slow motion, you know. And otherwise, I'm available. You can come and talk to me. My office is in the first floor here in the Dean's Suite. So thanks for your attention. And I'll stick around afterwards for those of you that don't have to go if you have other questions. But at least I give those who are on a tight schedule an opportunity to leave now. So thank you. And there's more handouts up here if you didn't get one.